We'll continue. After the stomach comes the small intestine. So the small intestine is probably the most important organ of the entire digestive system. It's the major organ of chemical digestion. Almost all of the nutrient foods, carbohydrates, fats, proteins, nucleic acids, are chemically um, digested here. It's also the place where the vast majority of absorption takes place, where these nutrients and nutrient molecules end up entering our body from the outside. Um, it's it says it's 2 to 4 meters long. So that's 14 feet long. They used to say it was far longer than that, but those measurements were all taken with dead people's intestines. Like, you don't really get to pull somebody's intestines out and stretch them out and measure them if the person's alive. They don't. They generally don't appreciate that. The problem is then when somebody's dead, there's no muscle tone, and the muscle tone actually can really shortens this up. So I think it's one of those. It's safe to say that it's irrelevant how long it is, but it's long, um, and that indicates how much surface area and how much time there is for absorption, because food moves fairly slowly down through it. Or, uh, yeah, it's divided into three sections. There's the duodenum, the jejunum, and the ileum. The duodenum literally means 12 because it's about 12 finger widths long, so about 12 inches. Um, and it's found retroperitoneally. The jejunum is and the ileum are both just have a dorsal mesentery. Um, it's here. Now, interestingly enough, the pictures, this is the duodenum, the jejunum, and then the ileum is here. The folds in here fold up like pages in a book. This is not random bunch of guts here. There is a pattern to it. Um, the other thing to remember is that what goes into the duodenum is very different than what is in the lumen here in the ileum just before it ends, just before the terminal portion of the small intestine. What enters here is chyme, and it's acidic, and aside from some proteins, it is pretty much not chemically digested. What exits here is everything that's really left over that we don't want to absorb, except for maybe some water. All the good stuff that we want has been taken out, and all of the stuff that we can enzymatically break down has been enzymatically broken down by this point. So it's not chyme anymore, but it's not yet feces here. So a lot of change happens over the length of the duodenum, or the length of the small intestine, rather. The duodenum itself, retroperitoneal, because it's got a couple of ducts going to it, the bile duct and the pancreatic duct. These two ducts enter the duodenum together at the hepatopancreatic ampulla. And there's a sphincter that controls that. It's called the hepatopancreatic sphincter, which is also known as the sphincter of Oddi. So it ends up looking like this. All the bile that's made by the liver passes down through these ducts, the hepatic ducts, and then down into the bile duct. Now, if the sphincter is closed, then the pressure of the bile backs up and goes through the cystic duct and into the gallbladder where it's stored, and we'll talk about in a few minutes. When we need bile, the gallbladder squeezes and bile comes out the cystic duct and then down the bile duct, enters the ampulla here, hepatopancreatic ampulla, out through the sphincter and squirts and mixes with the chyme here. Similarly, the pancreas has got this duct that runs the whole length of the pancreas, and pancreatic juice, which is full of enzymes, digestive enzymes, comes out here. Pancreatic juice is also very, very alkaline. It is exactly as alkaline as the chyme is acidic. So it neutralizes the acidity here. And again, this bile duct, or this pancreatic duct, enters the hepatopancreatic ampulla and then squirts out through the sphincter of Oddi to mix with the chyme. Now, this accessory pancreatic duct is open all the time, and it's for pressure relief. And so that if there's too much pancreatic juice, it's not going to build up in here. We'll talk more about this later. Retroperitoneal, so all of this doesn't move. It is kind of C-shaped, and it goes the exact opposite to the stomach, which goes around this way. So right here is where the pylorus of the stomach and the pyloric sphincter enter the small intestine. So throughout the entire small intestine, there's modifications that actually increase the surface area so that we can maximize absorption. First thing that you'll see is that there are circular folds. They're called the plicae circularis, which really means the circular pleats. The small intestine looks basically like a vacuum hose or an accordion, and that gives surface area. Now the surface of this is not smooth. It's more like terry cloth. It's like a towel, and each one of the little fingers of the terry cloth or the towel is called a villi, which means finger. And then each one of the villi is covered in epithelial cells that have a corrugated or finger-like surface of the cell membrane, and those are called microvilli. It's sometimes called the brush border. So basically, what this is like is a scrunched up towel, with the circular folds being the scrunching, the terry cloth being the towel. And as you know, a towel with deep terry cloth is more absorbent than a bed sheet, because there's more surface area. It's as simple as that. So the plicae circularis are permanent. They're about 1 centimeter deep, and they spiral and they force the chyme to move slowly. They provide almost friction, so it looks like this. Now, in a dissection, when you touch it, it feels like velvet because of the villi all on the surface. The villi are finger-like extensions. They're only about 1 millimeter high. And the surface of each villus is got a columnar epithelium, and it's a simple epithelium for their absorption. So these are called the enterocytes because entero means intestine. Now there's also goblet cells in this that create some mucus for lubrication. And so each enterocyte has microvilli on the surface. And intrinsic proteins, part of the cell membrane, the lipid bilayer, some of these proteins are enzymes, and we call these the brush border enzymes. And this is where the final chemical digestion takes place in these brush border enzymes. They do not mix with the food, but rather the food comes in contact with them. There's an enzymatic chemical reaction. The food stuff breaks down, is chemically digested, just seconds before it's absorbed. Now, at each one of these villi, at the bottom of it, there are these things called intestinal crypts. And these are where the epithelium grows. You can almost picture that the tip of the villi is the oldest. It's going to get worn off as food goes by. The newest ones are down protected in these little crypts. So these are stem cells that are making new epithelial cells. There's also there's lymphocytes that are useful for killing things that shouldn't be there. There's cells, pant cells that secrete defensin and lysozymes. There's enteroendocrine cells that are hormonal. They're part of the endocrine system. And there's cells that secrete something called intestinal juice. They're secretory cells. So when we look at a picture of it, it looks like this. Each villus, which means finger, has got these columnar epithelial cells with goblet cells in them. There are stem cells down at the bottom that are making new ones. There's enteroendocrine cells. There are all of these cells in these little crypts. And the crypts are actually like this to protect these cells. These cells are replaceable. These ones are harder to replace. Now, up the center of each villus is a venule, an arteriole, and a lymph vessel, a lymph capillary. And this is where the absorption takes place. Out here is outside of the body. When things cross through these cells and into this space, into the interstitial fluid, they are now absorbed into the body, where they enter the bloodstream here. Oxygen-rich, nutrient-poor blood comes here, goes through these capillaries, where it picks up nutrients, and nutrient-rich venous blood comes here. And it ultimately ends up in the hepatic portal vein, not general circulation. The lymph capillary is here to pick up things that are too large or not water soluble and can't get into the venules. So this would be picking up dietary fats primarily. Now, each cell, as you can see, each absorptive cell here in the epithelium is a columnar cell, and the surface of it looks like Bart Simpson's hair. And that's called the microvilli. Again, all for increased surface area, more absorption. Now, just below all of this, right there, you'll see there's the muscularis mucosa, and then this is the submucosa. Now, the submucosa has this lymphatic tissue where we protect against bacteria and anything that might be entering the body. Here, we catch them right away. So this is reticular connective tissue that it basically act like lymph nodes, except instead of dealing with lymph, that's dealing with interstitial fluid right at the site of absorption. There's also these glands called Brunner's glands. But they're only found at the beginning. They're only found in the duodenum, and they secrete an alkaline mucus that protects the duodenum from the acidic chyme until the pancreatic juice can actually neutralize it. These Brunner's glands are very important. When they stop doing what they're supposed to do, you can end up with a duodenal ulcer. The intestinal juice is secreted when the walls of the intestine, the small intestine, are either stretched by the presence of food, or they're irritated by the chemical properties of the food, the acidity or some of the chemicals that are in it. It's a little bit alkaline. It's mostly water and mucus. And the whole point of intestinal juice, people mistakenly think that intestinal juice is about digestion or even mechanical digestion. It's not chemical or mechanical digestion. It's to facilitate propulsion. It's basically lubrication. It's water and mucus. So it gives the water, the extra water added, gives something to dissolve the nutrient chemicals in to facilitate absorption. The water and the mucus make the stuff easier to move through the small intestine, so it's facilitated by that. Now, interestingly enough, this stuff gets recycled because the water and the proteins in the mucus end up getting reabsorbed. The proteins themselves, the mucin, gets digested, and the amino acids get reused. So this is a zero sum. There's no cost to the body to create this.